Hi, my name is Connor Howington, and I'm a summer research intern at the University of Notre Dame. I'm in the Data Intensive Scientific Computing Program, or DISC, an NSF-sponsored research experience for undergraduates. I've been working in Notre Dame's Bioinformatics lab, and I want to give you a brief look at the sort of work I've been doing this summer. My project is in a subfield of bioinformatics called computational genomics. Genomics is the study of the whole genome of a species. That is, it looks at all of the chromosomes that make up the species' DNA as a whole. At Notre Dame's Bioinformatics lab, we are interested in learning more about the genomes of a couple of important species that are involved in the transmission of malaria. This widespread disease killed a total of 425,000 people in 2015, and there is currently no effective vaccine against it. Having an understanding of the genomes of malaria-causing parasites and the mosquitoes that transmit these parasites can help us design drugs and insecticides to combat the spread of this disease. Meet Anopheles fenestis. This species of mosquito is one of the most common malaria vectors. However, our current assembly of its genome is much lower quality than we would like. Our lab has designed an algorithm called HESL that can correct errors in a genome assembly. Running error correction on a dataset as large as the fenestis assembly, however, takes a significant amount of CPU time. This summer, I worked on parallelizing HESL, which involved breaking it into a series of small, independent tasks and running it on a computing cluster that can take a bunch of tasks and run them at the same time. On the other hand, we are also interested in studying the parasites that cause malaria. We focus on a species called Plasmodium falciparum, which is responsible for the majority of malaria-related deaths. One particular strain of P. falciparum, called 3D7, has been the focus of the majority of laboratory research on the species. As a result, we have a pretty complete picture of its genome and a lot of information about it. However, often we want to analyze other strains, such as drug-resistant strains or ones that are isolated directly from infected patients, using the information that we have on 3D7. So this summer, I developed an algorithm that can create a mapping between two different genome assemblies. Using this algorithm, I was able to map specific positions on a drug-resistant strain of P. falciparum to genes on the 3D7 strain, and show that these genes corresponded to important metabolic pathways in the parasite. By studying these two key players in the malarial transmission cycle, the Notre Dame Bioinformatics Lab is helping to discover new ways to fight this disease and create a healthier, safer world.